Hello everyone. Our today's topic is antibiotic, in which beta-lactam antibiotics. It is a broad spectrum of antibacterial action. Now its mechanism of action. The lethal antibacterial action is due to the selective inhibition of bacterial cell wall synthesis. Specifically, it inhibits the biosynthesis of peptidoglycan, which provides strength and rigidity to the cell wall. Means it interfere with bacterial cell wall synthesis and bacterial cell division. Also, they act by inhibiting the synthesis of peptidoglycan, and peptidoglycan is an important component of the cell wall. Now, structure of penicillin was established in 1945 by Dorothy Hodgkins by the use of X-ray. Now on the screen we can see some structures of penicillins and its R group and drug name. First one is penicillin G, in which benzyl penicillin is attached to the structure. Next, penicillin V. It's also called phenoxymethylpenicillin. Next, ampicillin. It is D-alpha aminobenzyl penicillin, in which amino group and benzyl group is attached. Next, amoxicillin, in which amino group and at the para position hydroxybenzyl group is attached. Next, methicillin. It's also called as dimethoxypenicillin, in which at the 2 and 6 position dimethoxy group is attached. Next, penicillin contains a bicyclic system consisting of a 4-membered beta-lactam ring fused to a 5-membered thiazolidine ring. The skeleton of molecule suggests that it is derived from the amino acids cysteine and valine. Properties of penicillin G. It is active against gram-positive bacilli, that is staphylococci, meningitis and gonorrhea, and many but not all gram-positive cocci. It is also non-toxic. The penicillins are amongst the safest drug known to the medicine. Not active over a wide range or spectrum of bacteria. It is ineffective when taken orally, since it breaks down in the acid condition of the stomach, and penicillin G can only be administered by injection. It means it is not taken by orally, it is only administered by the injection. Sensitive to all known beta-lactamates. These are enzymes produced by penicillin resistant bacteria which catalyze the degradation of penicillins. Allergic reactions are suffered by some individuals. There are several problems associated with the use of penicillin G. Most serious ones are following. First one is acid sensitivity. Second, sensitivity to the penicillinase. Third, an narrow spectrum of activity. Now, structure, activity and relationship of the penicillin. Fused beta-lactam and thiazolidine ring forming a bicyclic system called penem. This bicyclic system confers strain on the beta-lactam ring. The free carboxylic acid group is essential. The strength beta-lactam ring is essential. It increases strength, increase in activity and increase instability. The acyl amino side chain is essential except for thynomycin. And sulfur is usual but not essential. The stereochemistry of bicyclic ring with respect to the acyl amino side chain is important. See stereochemistry of the hydrogen. Now on the screen we can see some structures of penicillin. in which amide is essential, cis stereochemistry is essential, lactam is essential, free acid is also essential. Very little variation is tolerated by penicillin nucleus. Also, any variation which can be made is restricted to the acyl amino side chain. Acid sensitivity of penicillin. There are three reasons for the acid sensitivity of penicillin G. First, ring strength. The bicyclic system in penicillin consists of 4-membered ring and a 5-membered ring. As a result, penicillin suffers large angle strength and torsional strength. Acid catalyzed ring openings relieves these strengths by breaking open the more highly strength 4-member lactam ring. And we can see this reaction on our screen is that it's ring opening. However, penicillin V is still sensitive to penicillinates and it's slightly less active than penicillin G. Ampicillin and oxacillin. Penicillin sensitivity to beta-lactamates. Beta-lactamates are enzymes produced by penicillin-resistant bacteria which can catalyze the reaction in which the same ring opening and deactivation of penicillin which occurred with acid hydrolysis. In this reaction we can see on the screen. Now, enhancement of gram-negative activity is found to be greatest if the hydrophilic groups, for example NH2OH, COOH, is attached to the carbon, that is alpha, to the carbonyl group on the side chain. Penicillins, which are active against both gram-positive and gram-negative bacteria, are known as broad-spectrum antibiotics. These are two classes of broad-spectrum antibiotics. Both classes have an alpha hydrophilic group. Class 1 broad-spectrum antibiotic, ampicillin and amoxicillin, where hydrophilic group is NH2 group.